Hello everyone, so I am here to record the video as promised because we were not able to have a good discussion time because of the internet issues and also the noise issues. So yes, it's here. We are now at the fourth SLHT, and this time it's the second topic, which is all about research design. So we're done with a review of the related literature, and now we're at research design. So first, research design is that defined as the logical and coherent overall strategy that the researcher uses to integrate all the components of the research study. So all your components are integrated there, and it is arranged such that you will be able to observe what you want to observe. Okay, in order to find meaning in the overall process of doing a research study, a step-by-step process will be helpful to you. Okay, but before we go to the step-by-step processes, let's familiarize ourselves with the basic concepts, the basic ideas behind research design. So first, let's go back to what is the scientific method. Any ideas? So you can pause this video and then comment or just try to rack up your brains on what scientific method is. So as you have remembered, we have iterated this in our past lessons, and scientific method is that process that science individuals or people who are doing scientific inquiry follow. It is a systematic set of steps that is followed, and then we can go back and forth. Is iterative, but there's a system wherein you ask a question, you find the problem, and then you do the background research, and then with what is known you construct a hypothesis, and then you test that hypothesis with an experiment, and then you analyze your data, and you not just present what is there, you analyze it, you draw your conclusions from that, and evaluate evaluated with the hypothesis that you gave, and then you communicate your results. So back here in the middle, this is the testing with an experiment. So for us to be able to have a good result here, a valid conclusion, let us learn the skill of designing our experiments well. So before we go there, let us try to have this example. How do we bake a cake? Okay, how do we bake a cake? So let's try to look at this time-lapse video from YouTube. So observe. So we're done here. So this video was actually a time lapse of a wedding cake video. And then in this video, we were shown with the process. First, they were preparing the ingredients, and then you have the eggs, you have the dough, and then you have the other steps that you created there. And so until the creator was able to have these layers of the cake, of the wedding cake. So when we design, so when we bake the cake, it is not enough that we know the ingredients. Now the flour, the eggs, it's not enough that we know how to beat the eggs. It is what we need is the steps, or there are other factors to consider for us to be able to come up with a good cake. So it's also the same with the research. It's not just we have the hypothesis already, we have the materials, the subjects, and the setups. We also must be able to know how to set it up well so that we can observe what we want to observe. All right, so let's go next. So here, when we want to have a good analysis, it is a rigorous analysis. A careful one would also require us to have a careful experimental design. So research, a good research really has its grounds on statistics, right? When we analyze it. And here now, we must be able to think of it, design it well, so that we will not be wasting our time. So if you don't spend time thinking about the types of observations that you'll be making, the likely outcomes, and how you can evaluate your data to differentiate statistically between outcomes, you might fail to carry out experiments that can actually provide you with informative data because you're not able to set it up well. Wala ka kahibalo sa imong padulngan, and that would be poor science and a waste of time and resources. Okay, so the experimental design, this research design, is the one that we're focusing on. So there are many types of research designs, and there are two types even. So the qualitative and the quantitative, and then at the quantitative, there are also many other categories, and under that is the experimental design, which our class is focused on because we are at the STEM research. Alright, so this research design is based on scientific method called the experiment. So we focus on experiment with a procedure of gathering data under a controlled or manipulated environment. So as much as we can, we try to control or manipulate that environment so that it will be a simulation of one variable causing the change in another without the effects of the other variables in the surroundings. So we must control it. So it is also known as true experimental. So naamatay daghan, right? Remember the quasi, the pre-experimental. So it's true experimental design since it applies treatment and manipulation more extensively than the quasi or the quasi-experimental design. So random assignment of the subjects or participants into treatment and control group is done, increasing the validity of the study. And experimental research, therefore, here in the last part, it attempts to affect a certain variable by directly manipulating the independent variable. Okay. So in order to let's try to read this first. Yeah. So to reiterate, the analysis of your paper has something to do with the statistics, right? And your statistics has something to do with your data. Okay. And your data is gathered from your experiment, the one that you built. So it must be designed well. You must be able to have a good design. Alright. So that is why it's very important to have a good research design. So when we have this in mind, we will have good results. And yes, good results to be shared with everybody else. Okay. So in order to perform meaningful analysis, In order to perform meaningful analysis, you need to design your experiments with statistical principles in mind. Yes. So we have this statistical principles, and this includes number one, accurately and clearly defining your variables in the sample space. Okay, what variables would you want to observe? And just how many people would be included? Just how many plants? Just how many cockroaches? Alright, and then accurately defining your factors and the levels of your factors. So, for example, your factor is you're trying to make an extract, so that is the effect of that extract. And you can have levels of factors. If the extract would be just 50% on concentration, 70% concentration, 100%. Okay. Next, identifying the type of experiment you are running, making sure that appropriate controls are used. Okay, making sure that you perform enough replications to create a representative body of data. So really, in a well-designed experiment, we have to make space for replications, because sometimes things happen by chance, and some effects seem to be different from the ones that we expect. So to have a good comparison, you must be able to replicate it also. And making sure you understand the likely distribution of your data, normal distribution, and ensuring that so that you will know the type of statistics that you will use, and ensuring that you are aware and familiar with the types of exploratory and inferential analysis used in your field of science. So in our studies, most of the type of tests that we will use are the analysis of variance, ANOVA, and the t-test, which we will be studying also in the future lessons. Okay, so we have to put our mind also with that statistical part, because if we just do the design right away without knowing what type of test will we be using, and then just to find out in your analysis that you need this data DI and you need to compare two means instead, and you just have one set, so yeah, nothing, mabalik jud ka og experiment. Okay, so to avoid the wastage of time and resources, you have to make sure you design it well. You must design your experiments with data analysis in mind, because if you don't think about analyzing your data until after your experiments, you're going to run into big problems from someone concerned. Okay, so the best way to get a handle on what your experiments are likely to entail, so you have to expect on what are the standards that other people are doing who are doing the same study as yours. So you look at papers in your area of science and see what other investigators are measuring and how they're doing it. So you take note of things like what variables they are investigating, which of the independent variables are, and which are the dependent variables, and what their sample size is, and meaning how many observations do they make, what controls they use. So for example, they use the control, what standards they use against, and then how many experimental replicates they have. Okay, so you look at this, and if you're able to find a study, you can even adapt their methodology also, and then do it with yours. Okay, adapt means adapt. There's a difference between adapt and okay, there's a difference between adapt versus adopt. Adopt, this one, you really just get it and copy verbatimly everything. So adapt is just you just look at some portions as a guide and then you use it. But still better to have to be able to communicate with the writers also. Okay, there are two types of variables. So because we are talking about variables in this part, there are two types of variables. So these are the quantitative and the qualitative variables. Quantity versus quality. Okay, quantitative variables are the ones that differ in magnitude. So they can easily be measured, recorded as a number. So number dyan siya. Examples of these variables are, you have the age, your height, your time, the weight. So the examples are drawn here below. You have also distance, mass, you have volume, all those quantities, physical quantities. So quantitative variables are easy to summarize using numerical calculations like the median and the average. Okay. After that, let's go to qualitative. So in a qualitative, referred to as the categorical variables by category are ones where the observations differ according to the kind. So in kind, qualitative variables can be placed in categories like gender, the male versus female, or the marital status, unmarried, married, divorced, widowed. This makes them particularly good for summarizing as percentages or in a pie chart. So in any sila percent. Sometimes qualitative data can be ranked. For example, a fruit survey. So, survey kasi mong friends on how a certain fruit tastes to them. They can have this ranking as very sweet, moderately sweet, slightly sweet, neither detectably sweet nor sour, slightly sour, moderately sour, and very sour. So you can have this as their choices 1, 2, 3, 4, 5, 6, and 7. But these are not numbers because these just are referred to this equivalence. Not really nga numbers, give nga numbers na quantity. Okay, so more closely, let's have this table. So for quantitative variables, we have two types, the discrete and the continuous. When we say discrete, here, let us read. The data are described numerically on a finite scale, and there is a logical limit to the precision. Finite scale, okay, you can count 1, 2, 3, and 4. There's no, you can have, for example, number of children in the family, bacterial colonies on a plate, coin toss, shoe sizes. So number of children, you can have 1, 2, 3, 4, 5, no 0.5. Discrete, defined, finite, naay ending. So bacterial colonies on a plate, coin toss. Coin toss, you can have once, twice, okay, and then each toss you have the probability of 1/2 because you can either have the head or the tail. So these are the common statistical tests, and summarizing the data, you can use the mean, median, mode, chi-square, standard deviation, and so on. For the next type, quantitative gihapon, continuous type of variable, continuous nga number. When we have the continuous data, they are described numerically on a continuous scale. They can be broken up into infinite measurements. So between 1 and 2, there are infinite numbers that you can divide. So there's 1.5, and between the 1.5, you can have 1.25, and then in between that, and so on. So you can get closer and closer to 0 as well. No. 0.5 and then 0.25. I'm sorry. So theoretically, there is no limit to the precision. But in real life, we have a limits depending on the instruments that we can use. For example, for the measuring of the length, you have the ruler. Your ruler can measure up to the 0.1 maybe of the centimeter, and so on. So you can have a more precise instrument, the vernier caliper, as a measure of length, and so on. So examples of these types of data, you can have the temperature, the age, weight, time, length. So nice, sumpa, you can have 1.1 meters, 2.5 meters, 2.51 meters, 2.512 meters, 2.1828 centimeters, 5.67 times 10 to the negative 8. You can have all those values. Okay, so common statistical tests and summaries, you have mean, median, standard deviation, standard error of the mean, regression, correlation. So here are the commonly used tests. Continuous. Okay, so discrete versus continuous. Now let's go to the qualitative. There are also types of qualitative variables. We can have the nominal and the ordinal. Nominal, the data are described by words or categories, and they are not numerical and cannot be automatically ranked from high to low. So you have colors, you have red, orange, green, blue. From you can go other colors like maroon, tangerine, burgundy, other colors that you can think of. Okay, so you cannot rank them. They're just an identity of another color, another color, and another color. Gender, you have male, female, and also the other categories in between. And then you have the occupation, other kinds of occupations. You can name them. And then the location. So there are words, categories, names, identities, and so on. And then you can use these tests, the mode, chi-square, chi-square. Yep, chi-square. And then the ANOVA also, and the paired t-test. Next, if you can rank them, just like our example of the fruit taste, these are called ordinals. So the data are described by words and categories as well. Although they are not numerical in the sense that the values can be added or subtracted, they just can be ranked from high to low. So the amount of pain. So remember that what's that movie with the dashi? There's the white one. Amount of pain from 1 to 10. Katong mangutana ba? Ang sige name to eh. Yeah, katong circle something like that. And it comes to 1 to 10. It's like a health machine. I forgot. Okay, and also the most scale of hardness for minerals. So the hardest one is diamond. The IQ number, and then the IQ number. So the IQ number, you have the IQ number, and then you can also have the agree or disagree nga scale. Strongly agree, strongly disagree. Ordinal gyapon na sila. You can have your survey pa check, or you can let them choose, or you can let them have one as strongly agree, two for slightly, like that. And then you can use median mode Kruskal-Wallis test, ordinal logistic regression. So we can study this also in another time. And then so those are your qualitative variables. Again, your quantitative, you can have continuous data, or the number is continuous. You can also have discrete. Okay, putol, 1, 2, 3, 4, 5. Counting, okay, counting. And then continuous is sumpay siyang data, temperature, and then age, weight, height, nana. And then for the qualitative, nominal or ordinal. And then now, after our types of variables, we can also have interacting factors in a design. Factors could also mean your variables. So the different choices are the variables in your design. So the goal of your experiment is to measure the effect of a particular variable or the set of variables on a system. Okay, you have to measure the effect, but how does the number of the interacting factors in your experimental system impact your design? Okay, so the most important step is first to look at and think about all the possible variables, or also called the factors, which might contribute to your results. Okay, so para ma-weed out nimo unsa jud imong focus, you think of all the possible factors. Okay. You want to test which brand of tires. For example, which brand of tires, A bakaron or B, results in the best gas mileage. So when you're driving on the highway, you first need to identify all the possible factors that you might that might influence the gas mileage. Mileage stands for the gidugayon sa madagan sa inani nga amount sa gas sa imong car. Okay? Talking about cars ni siya. So unsa kaya kalay-a ang maadto niya? Okay, so you have many factors to consider. You can have, what factors could they be? Number 1, the type of car. Yes, of course. And then, because you want to naapoy weather, because maybe the weather is not good for the day, and then hindi maayo ang visibility pa karon sa kalsada, nanana. Or also the type of road. Kung bumpy road ba siya or smooth lang siya nga road. Okay. So mo ito yung nahunang-hunaan nga factor. Wear and tear sa road. And then also the speed. Okay? So unsa kaya? So kung speed, kung unsa kaya maayo sa mga tires. So you just want to find out which brand of tires are better. So after that, you have identified your factors. You will then design a fair test. What is a fair test? A fair test is a test which controls all but one variable when attempting to answer a scientific question. Only changing one variable allows the person conducting the test to know that no other variable has affected the results of the test. What does that mean? Okay, in a fair test, what's going to happen? A fair test is that type wherein all the others ang imong gi-control para jud ang imong gi-observe ra jud na variable ang makita. Okay, so atong car, ang atong gibuhat is ang imong car. I'm sorry for the drawing. I'm not good with drawing cars. So there's a tire here. So laylay mong tire. So ang usa nga setup, the same car. But you can have brand A for one car and another car na pwede mo same car. But this time you use brand B for the tire. Okay, and then you just you test that one. Imo ba siyang parehason ang imong, aside from these changes here in the variable sa imong tire, tanan imo parehason, same car, all the other variables will be the same para fair. That's why it's called a fair test. Okay, however, there are many times when more complex experimental system, there are sometimes when it's not just the variable nga tire ang makaapekto, and then you would like to have a more complex experimental system where you need to assess whether either the impact of several interacting variables on the final outcome is seen. So makita ba ang effect, and then or it is prohibitively expensive lang ba jud or physically impossible to change one factor at a time lang. So a fair test is outlined above will tell you which brand of tire resulted in the best mileage for a particular car. The one you used to do the test, under very specific condition. Katong atong i-drawing ganina, you can have that as one fair test. Okay, but we can have, what if you wanted a more general answer to the question of which tire brand results in the best mileage, one that was applicable to different cars and conditions. So you might suspect that the best tire depends on the type of vehicle in question, pwede ba minivan, a sedan, or a pickup truck, or how worn down the tires are, kung bago bang imong tire after 5,000 miles of wear and tear. Okay, so it increases then the total number of fair tests required. As the number of factors and the number of choices per factor increases, the number of fair tests needed becomes prohibitively prohibitively large. And when you have many factors and are choices per factor, it is necessary to use a different experimental design. Nani sila gitawag ng mga orthogonal arrays and factorial analysis, which is beyond our subject for me to discuss to you. Okay, what this just mean is that, for example, usara atong gitest, number of factors to test, and then if, for example, this is just the tire, okay, and then the number of choices per factor is A lang, tire A versus tire B. So you just do same car, sorry, wrong, same car, you just have that, you can have here. Okay, so here, so just have one factor, the type of car, A versus B. So 2 experiments. This is 1 experiment and 2. What if more factors, your tire brand pa nimo, car type, wear and tear kung new versus old. So you can have this experiment, and what I'm showing here is that you can have tire brand A, para sa mo car type A, dayon tire brand A, imo jud pair tanan para i-compare. Tire brand B, nya parisan pa jud nimo bag-o nga A versus old nga B pa jud. So ansa sa car brand B na pod ay tire brand B nya car type A, and so on. So you can have how many threads now? How many experiments? So theoretically that would be 1, 2, 3, 4, 5, 6, 7, 8. You can have 8. Just like this one. So here, if 3 factors atong gitest katong car brand, car type, unya wear and tear nya, duha-duha lang man kapilianan per choice. So 8 ka buok experiments imong buhaton. So 8 ka murag pares-pares. So that's the theoretical number of fair tests that you need to do. Unta, wala pa'y apil ang imong replicates. So mao na nga, the more choices you have, the more levels pa jud. For example, ang kani kay ato ang choices kay pwede na siya ma in exchange for concentration. So for example, you want to do an extract, moringa extract. Let's just do it. Moringa extract. So ang imong moringa extract, this is your factor. Only one. But your choices, you have 50% concentration, 75%, and then 100% concentration. So mao na nga, you can have how many experiments? Only one factor. So you can have one experiment na kani mong gamiton. Like, for example, asa ba ito ibutang ang extract sa imong one setup nga mga cockroach. So this is experiment 1, and then you can have another the same setup, but this time 75%, and then have another setup. This time it's 100% extra. So 3 experiment. 1 factor, 3 choices. 3. Pero kung duha mo factor, like extract nimo kay lain plus another pa jud na extract nga kuan nga separate siya nga extract. So muna nga gamiton to ni mga table here. So katong atong ibuhat na example kanina kay unsa man to? 1 factor, 3 choices. So kani 3 experiments, 3 fair tests, controlled experiments that you need to do. So understandably, ma-solve na siya gamit ang mathematics, combinations. Okay. Then creating well-controlled, you can study this branch of math kani pag solve ani sa combinations. Search lang man ang combinations. Okay. Then creating well-controlled experiments. So when doing your projects, okay, regardless of whether you are conducting experiments to evaluate one or many factors, you will need to design a well-controlled experiment to give you that fair test. So you have controls that will allow you to, number one, evaluate on a technical level whether an experiment is working. So, ito musulod ang mga controls. So, the controls help you do this. Number two, help you to interpret the results by giving you standards to compare against. Number three, guard against unforeseen factors that might bias your results. For better and for worse, the word control used by researchers are given several but related meanings. Okay, so to summarize the different usages, okay, we have this table here. So first, let's go to the control group. When we say control group, okay, it's an experimental trial where the independent variable is set at a pre-selected level, often the variable's natural state, for the purpose of comparing to all other experimental trials. So, that group muna siya ang imong gibutang para makumpara nimo sa imong experimental trials. Understood? And then the second word, let's go here first. Controlled variable, quantities that a scientist want to remain the same between trials, so that the effects of only the independent variable are being measured, sometimes called the constant variables. Now let's go to the positive control. So one or more experimental samples which are known from previous data to give positive result in the experiment. A positive control is used to confirm that the experiment is capable of giving a positive results. Positive meaning makita, observable results. Not positive, negative, good or bad. Positive, makita. There is an effect na iyang mabuhat. Whereas, negative control, one or more experimental samples which are known from previous data, from previous researches, to give a negative result in the experiment. A negative control is used to confirm that the experiment is capable of giving a negative result. So, most of this one refers to no effect. So, such as, kanang water lang imong ihatag, or wala jud, negative control. Positive control, such as butangan dyan mag standards. We will see in some examples. Number one, positive control. So you have here, you're designing, so in the reading, let me have the reading. In the reading, ang example dito kay, unsa man to, kadtong mga light bulb design, or that was designed for mga circuits. Okay, so if you conduct an experiment, and see that your positive control behaves in an unexpected manner, you have the cause to doubt the validity of your experiment. For example, if your research question was, will this new circuit design work to turn on a light bulb? So, maka-turn on ba daw ang imong circuit design? So, imong buhaton kay, imo sad i-confirm kung tarong ba imong mga light bulb. So, magamit ka o sa first imong design, here, ipa-turn on sa nimo imong light bulb. So, para maka-confirm nga mo, siga jud ay kay base di ay og daot imong light bulb. Nya, pagbuhat na din mo sa imong design, ay, example kani imong design sa circuit kay kuan day siya, kanang mapalong day siya. What if daot, for example lang, kaning start here. What if daot day imong light bulb? So, dili sa jud busiga bisag unsa pa din imong design og tarong diha. So, muna ang gitawag na false negative. Meaning, negative result kuno, pero false rada ito. Kay daot jud imong light bulb. So, you must have this positive control nga mapasiga sa ang light bulb. Ordinary design. So, for example, wala lang ni sa middle. Okay? So, this is to control, to avoid a false negative result like this. So, you kailangan ka og positive control. So, In other words, pwede ka maghimo og another forms like lain pa karon light bulb, basta kanang ang light bulb nimo sure jud ka nga mosiga siya. Okay. So just as positive control are used to minimize the impact of false negatives in an experiment. Negative controls this time. Okay. The negative controls are used to minimize the impact of false positive. So, positive pero sayop dahil na positive. Okay. So, meaning, for example, false positive, magsiga ang suga pero dili dahil ang imong design more cause ato. Okay. So, negative controls confirm that the experimental procedure is not observing an unrelated effect. So, for example, ang negative control nako kay kani. Gikuhaan ako siya og dire sa middle. So if I really get this one, meaning ang ako light bulb mopalong jud na siya. Mopalong. For example lang wala na siyang mga light ya. So mopalong jud ako light bulb, negative control. So meaning dili siya mosiga, meaning wala jud other hidden nga mga light source from other circuit nga wala gipakita nako sa diagram. So, that one is a negative control. Ato jud ipa-show nga dili jud siya mo work ani nga circuit ta. And then if ato na dayon buhaton sa atong design kay mo siga na jud siya. So, para maka-avoid ta og unsay tawag ato false positive. Pareha gud sa mga kuan kanang na sila giingon nga kanang mo positive daw ka sa mga COVID sample test kit na. But if daw false siya nga positive, so na-declare ka nga positive pero dili di ay. So we do not know, di ba? Okay. But then, yes, stay safe. Amping-amping og follow the protocols. Okay. So again, your negative control lets you avoid the false negative. And then negative control lets you avoid the false positive. Okay. Other examples, we have this experiment. So your positive control group, 1 ounce of known fertilizer and water, and then your negative control just water. Then here, experimental group, 1 ounce of experimental fertilizer and water, and then your experimental group number 2, 2 ounces of experimental fertilizer and water. Kani siya, for example, known na dyan siya na fertilizer. Unsa na dyan brand na known. So, muna ni ang standard. Then, kani experimental lang ni siya na fertilizer. Bali, hinimo ni mga fertilizer made out of paper. Baka ron, 1 ounce niya. Kani kay, lahi na po na level, 2 ounce. So, imong gi-vary ang level. So, positive control it with the ones nga muhatag og effect na jud. Alam na jud nato nga mo-effect jud ni siya nga known na fertilizer kanang NPK baka ron or na siya nga brand and so on. Negative control, wala jud siya fertilizer, water lang. Okay. So constant po ng water sa tanan naman for water. Okay. So positive control, a group in an experiment that receives a treatment with unknown results. Nabalo na ta ani nga mo-effect jud ni siya. Negative control, example your control group kay wala di siya gitagaan o kanang tambal or vaccine pa karon saline shot ra siya. So kuan siya na salt ra. Okay, so other examples. So there are other types of studies nga dili ra ka makakuan nga positive control or negative control. So more than a simple yes or no. So it's often used with standards to compare the test samples against. So standards are products or practices in a particular field that are controlled or they're collectively thought of as working well. For example, mga standard na dyan vitamin C or standard nga tambal sa mga headache, for example, aspirin ba kay dili no, just mga standard ba karon nahibaw-an sa the doctor ang doctor na nahibaloon sa nga standard, so you can also research food. So going back to your experiment kaniha katong new circuit. So if the question wasn't just whether the new circuit resulted in powering the light bulb, but also how efficiently it did so, would be useful to know the amount of power used by the new circuit. So kung efficient na lang, pwede i-test na to ang power pod. How much kurente ba yung need over time. Okay, so not in some types of research, we have this example here. Determination and comparison of vitamin C content from Moringa oleifera by different methods. So, kani siya nga study, compare dayon niya kung how much ang levels of vitamin C sa Moringa oleifera. Iyan ang gilay-lahi ang methods. So, nagkuhan siya mga fresh leaves are analyzed to have content for ascorbic acid niya. Iya din compare it with other nga mga levels na known sa vitamin C. Okay, so iyang gikuha na. Was found to contain 0.82 milligrams per gram of vitamin C, which is roughly 2 to 4 times higher than that of from known sources of vitamin C, vis-a-vis such as kaning mga lemon and Mozambique. Okay. So, compare niya sa standards. So, creating a well-controlled experiments. There are some research, we're done with this. Yes, kani siya mga standards. Oh, there's some research nga dili jud buhat pud ang standards, ang positive or negative controls. So, this is often the case with sociology experiments. So, kaning mag-involve siya og preferences or behavior of humans or animals pud pwede. So, in this case, it is often you employ a control group to compare your test or your experimental group. Okay, for example, headache, you deal with people, and sometimes the headache is ma-feel man jud na dili man na siya ma-observe jud siguro. Dili siya dito visibly ma-observe. So, you can have persons, you can interview people if na-alleviate ba ilang pain. So, sa imong hypothesis, your aspirin works to alleviate headache. So, naka-control group. So, ang control group, kung na sila headache, imo silang, when they feel nga nagka-headache sila, imo silang dili tagaan kay control group man. And then, for experimental group, imo silang tagaan og aspirin sa tambal. After day ana, imo dayon, butangan ba karog effect time, na mag-effect ang tambal. After ana, imo din sila pangutan-on kung ni gana ba or unsa ilang na feel. I-compare nimo ang na feel sa control group versus sa experimental group. Okay. So how about controlling biases in experiments? So we have this called mga blinds. So not a single blind or double blind. So critical information is kept from the test subjects, but the experimenter knows everything. Single blind experiment. So for example, kani nga person kay tagaan na ni siya og placebo nga tambal. And then kanino siya kay naadyoy butang treatment. So wala pud kay balo ang ni appeal sa experiment nga na ba koy tambal or wala. So blind ang tao. This goes with mga consent pud jud. So, single blind experiment. Next, double blind. Ang mga test subjects o kang experimenter, wala jud kay balo. Kaya basin ay bias pud. The way they treat the people, their interviews or in their observations, mabias man pud kay balo ko nga placebo ra niya kung gihatag niya kang usa kay na jud treatment. So, mabias man tamo nang naay gitawag nga double blind experiments. This is very tasky pod and it needs a third party also. So the information is only revealed to the experimenter once the data has been collected. Okay. Okay, so we're done with reviewing that article. It's a long one, and then those are the fundamental things that you need to learn about the basics of research design. After that, we can go focus with some research designs put, and we had studied about these in the first quarter, katong different types mga pre-experimental, true experimental, quasi-experimental. And if we still have time, we will go to, if you can still remember third year ninyo or grade 9, katong ganing randomized design, completely block designs, inato nga mga design. We will try to have that also in the next lesson. So just read and read and read more. And hopefully you got some of the you'll able to finish until this part of the video, and then if you have any questions, just you can leave a comment, you can give me a message, and then we can answer them together. Daghan jud why jud kayo ni siya nga topic ang research design, and it's for now it's good for you to just familiarize and maybe not dive too deep lang po siguro, and then we just use what we need for our study, because even I is not dili ko knowledgeable sa tanan jud na research design, especially sa kaning mga scientific jud kayo nga research design. And that's why we can ask other people as well in designing our research design. What's important is that we know the principles, such as making good controls, and then designing it well, and removing biases and other things pa good to consider. Okay, so moreover, thank you so much for reaching at this part of the video, and see you next meeting. Thank you so much, everyone, and goodbye.